So we talked about catabolism through central carbon metabolism. We talked about the anabolism through mostly synthesis of amino acids, but also synthesis of fatty acids, synthesis of nucleotides, etc. These are also anabolic reactions. There is one very crucial point in making a metabolic network, in coming up with a metabolic network that can be simulated. And that is the addition of, incorporation of a biomass reaction to the list of reactions that you have compiled. And this is very important, and I will spend some time in sharing with you the idea behind biomass reaction and how it can be constructed. It is also referred as growth reaction. So you already know that we can represent metabolism as a set of metabolic reactions, right? We can write those reactions as a list. Here the idea is, if we have a microorganism, you know, microorganisms, they grow, right? So how do they grow? They consume glucose. Then there's lots of reactions taking place within the cell. And then at the end they grow, right? They divide, for example, there are new cells, etc. This is only possible because they consume some substrates, such as glucose. So, can we represent the growth of a microorganism as a reaction? So this is the question asked, and the answer is yes, you can construct a biomass reaction. So we are trying to write a reaction which can define the growth of microorganism. Why is this important? Because if you can write a biomass reaction, if you can represent the growth of microorganism as a reaction, then you can use this biomass reaction together with all other reactions in your system, such as glycolysis, amino acid synthesis, etc., all those reactions. You can use this list of reactions now to simulate growth behavior of your microorganism. How we will talk about this, of course, within this course in the coming weeks in a very detailed way. But incorporation of biomass reaction enables you to simulate growth behavior of microorganisms. So let's say you have your fermenter, you are growing your microorganism here, you have already put some glucose inside. Let's say this is the consumption rate of glucose by your organism, 5 millimoles per gram dry weight glucose per hour is consumed by glucose. So the question is, if you have this information, and if you have this list of reactions that represents metabolism, including biomass reaction, can you make simulations to predict the growth rate of the organism? And the answer is yes. So that's why these metabolic models are really important. You can use them, you can use these lists of reactions to predict, for example, the growth rate of your organism by using the rate of consumption of glucose by your organism. So we'll talk about this step later in the coming weeks. Since biomass reaction is very important to make predictions, now today I will talk about how this biomass reaction is constructed. So here we see a list of reactions. Some reactions are from glycolysis, Some are from pentose phosphate pathway, some are from TCA cycle. You see the names of the reactions themselves, right? Lactate is, and pyruvate, for example. Pyruvate is used together with an ATP to make phosphenol pyruvate, for example. And you see here the associated enzyme EC numbers. Here you see the associated gene names. So you have this metabolic reaction list. So you already prepared your list of reactions for relevant pathways, and you want to add a biomass reaction to the list. And as we said, this will help you to simulate and predict growth rates of your organism, your microorganism. So that's why I'm talking about this biomass reaction. So in order to give you more insight, I will talk about macromolecules within the cell first. So the cell is composed of 4 different major macromolecules. These are called macromolecules because these are really big molecules. It's not like they have 3 carbon, 4 carbon, etc. They have hundreds of or thousands of carbons with themselves. They are big molecules. So there are 4 major macromolecules that makes the cell. One is lipid, the other one is nucleic acids, DNA and RNA, the other one is carbohydrates, and the other one is proteins. For Saccharomyces cerevisiae, for example, a Saccharomyces cerevisiae cell is composed of 40% protein, 20% carbohydrate, 7% about nucleic acid, and 5% lipid. So this composition can be different across microorganisms or across other organisms. But roughly, we have a high pool of protein and a lower pool of nucleic acids usually, and we have a pool of lipids and carbohydrates. So we know this. Experimentally, this can be measured. What is the content of macromolecules within this cell sample? This can be quantified. This is the starting point to write a biomass reaction. So if I know this, then can I write the formation of cell as a reaction? Cell is composed of lipid, nucleic acid, carbohydrate, protein, and of course energy is required during growth, right? Some energy consumption, and this would make my biomass, my cell. And the coefficients of those reactions are the percentage content of those macromolecules within the cell. So this is how I start incorporating growth into metabolic reaction list. First, I write a biomass formation reaction in terms of the relative content of macromolecules. Here you see the content of Saccharomyces cerevisiae cells from Metabolic Engineering Principles book. So roughly these are the percentages reported there. Just carbohydrates, it's a little bit higher. And here you see the energy required to make those polymeric structures, those big molecules. So per gram cell, 24 millimole ATP is required for the synthesis of those macromolecules. So which means that here you should add, instead of energy, you should add 24 ATP. Just from other sources, I have shared with you the macromolecule content. As I said, these are quantified, measured by others. So for again Saccharomyces cerevisiae, you see that it has the cellular content is 45% protein. These are carbohydrates, about 40%. We have nucleic acids, about 7%. We have 3% lipids. Now I will define a term called building block to link macromolecule-based biomass reaction with the reaction list we already have. We have already compiled based on central carbon metabolism reactions and anabolic reactions. So each macromolecule is made from building blocks. How? A protein is actually an amino acid sequence, right? Amino acids are bound together to make proteins. So if you zoom into any protein, you will see amino acids. You will see alanine, aspartate, glutamate, leucine, etc., the other amino acids. So protein is a macromolecule, and amino acids are building blocks. So amino acids are building blocks for protein macromolecule. What about the other macromolecules? For nucleic acids, RNA and DNA, they are made up of adenine, guanine, cytosine, thymine, and uracil nucleotides, right? So those nucleotides are building blocks for nucleic acids. For lipids, fatty acids and phospholipids are the building blocks. So fatty acids and phospholipids, they come together to make lipids. And for carbohydrates, we also have other smaller molecules that come together to make carbohydrates. So these are called building blocks. Indeed, I will come here, but this is the link. So from glucose, metabolic precursors are made. Then metabolic precursors come together within a series of reactions to make building blocks, amino acids, fatty acids, phospholipids, nucleic acids, nucleotides, etc. And then those come together, for example, amino acids come together to make protein, to make macromolecules. So this is the relationship between metabolic precursors, building blocks, and macromolecules. For example, acetyl coenzyme A is a precursor within central carbon metabolism. It is used to make fatty acids and phospholipids, which are building blocks, which are later used to make lipids, macromolecules. Or alpha ketoglutarate oxaloacetate, they are metabolic precursors from TCA cycle. They are used to make amino acids, which are building blocks, and later amino acids are used to make proteins. So let's go back here. And remember, we had written already a reaction here to represent biomass formation from macromolecules. Now we will write similar reactions to represent the formation of macromolecules from the building blocks. So if you isolate protein pool from a cell, you can again, by using experiments, you can detect the relative content of alanine, aspartate, glutamate, serine, etc. within this protein pool. So this protein pool includes hundreds of proteins within the cell at that moment. And then you can quantify, as I said, the relative content of amino acids within this protein pool. Since this is available in literature for organisms, then you can write a reaction to represent formation of protein macromolecule from its building blocks. So this protein in this specific organism, let's say, includes 4% alanine, 5% aspartate, 15% glutamate, etc. So we can write a reaction to represent its formation from its building blocks. Similarly, we can write a reaction to represent the formation of lipid, a macromolecule, from its building blocks, fatty acids and phospholipids. Similarly, since RNA is composed of adenine, uracil, guanine, and cytosine, we can write the relative content of an RNA molecule in terms of adenine, uracil, guanine, and cytosine. So we can represent RNA macromolecule formation from its four building blocks as a reaction. So a sketch from this paper. So we have organism, we have macromolecules, we also have carbohydrates. It's not put here. So as you see, the biomass formation is written in terms of relative content of protein, RNA, etc. And then the synthesis of macromolecules from building blocks is separately written as a reaction. So RNAs are made up of, so their building block is nucleotides. So some fatty acids are building blocks for lipids. Amino acids are building blocks for proteins. And we write the reactions. And this is measured data from literature, again from Metabolic Engineering Principles book. As you see, for different microorganisms, they report relative amino acid content of their protein pool. So you can use these percentages to write a protein formation reaction for those organisms. And another example from human, cancer cells also, tumors grow similar to microorganisms. So if you want to model the tumor metabolism, you can also compile the metabolic reactions that take place within tumor cells. And if you want to simulate this, if you want to simulate tumor growth behavior, for example, you can also make up a biomass reaction for tumor cells, add it to your list of reaction to make further simulations. So indeed we did this with a student of mine and we published the results in 2016 in the journal in Neuroscience for glioblastoma, a tumor type for brain. So we made literature search first to identify relative macromolecule composition of tumor cells. Sorry, this was, I think, brain cells, for the brain cells, yeah. And we saw that about 40% is protein, about 55% is lipid, about 4% is RNA, and carbohydrate content is negligible. So we used this information to write a biomass reaction. Where will you see it? Here. You see, this is our biomass formation reaction. We also included the energy required to make those macromolecules. Again, from literature, we have found the amino acid composition of for mammalian cells, lipid composition again, and RNA composition. So we have also written reactions for the formation of protein macromolecule from its building blocks. RNA from the building blocks and lipid from the building blocks. So at the end we have 4 reactions. One for the synthesis of biomass from the macromolecules and 3 for the synthesis of macromolecules from the building blocks. If we had carbohydrate, we would have one more reaction here. This time synthesis of carbohydrate macromolecule from its building blocks. So we would have 5 reactions. And these 4 or 5 reactions are enough to link growth behavior with the reaction list we have compiled. Because in the reaction list we have already compiled, we already have alanine formation reaction, methionine formation reaction, or cholesterol formation reaction, arginine formation reaction, phosphatidylenoacetal formation reaction, the formation of those nucleotide structures also. So we already have all those formation reactions, and by adding those reactions, we link all those reactions to the synthesis of macromolecules and production of, the growth of the cell, the biomass reaction. Here you see a table from this paper. This paper is the first genome-scale metabolic network representation of Saccharomyces cerevisiae. And here you see how they made a biomass reaction. So here you see amino acids. Here you see the building blocks for RNA, AMP, GMP, etc. And here you see building blocks for DNA. Here you see the building blocks for lipids, building blocks for carbohydrates, and energy to make biomass. So indeed there are 3 ways of representing biomass reaction in metabolic network models. One is, we have already talked about, add 5 new reactions to your already compiled list. Why 5? So you will add 1 reaction for the synthesis of biomass from macromolecules, and for each macromolecule, lipid, protein, carbohydrate, and nucleic acids, you will add their synthesis from the building blocks. So in total, you will have 5 reactions. So if you add those 5 reactions to your reaction list, now you are ready for making simulations. Another approach used in the literature is, rather than adding those 5 reactions, combine those 5 reactions within a single reaction. So combine all 5 reactions and write them as a single reaction. So this means that you will write a single reaction of biomass synthesis from not macromolecules but from building blocks. I will show you how. And there is a third alternative, but before the third alternative, let me show you some examples. Are they here? Let me see. No. So let me switch back to this slide to show you how you can write a single reaction rather than 4 reactions, for example. So it's very easy. Here we have a coefficient for protein, and this is the protein reaction. So you can put this reaction here indeed, and multiply all coefficients here by this coefficient. Then you will incorporate, protein formation from its building blocks into the biomass formation reaction. The same here, we have lipid here, and this is the lipid reaction here. So you can put this reaction here by multiplying all coefficients here with this coefficient here, then you will write, so you won't have lipid explicitly in your biomass reaction. Rather, you will have lipid building blocks in your reaction. The same with RNA, you take this reaction here. If you have carbohydrate, the same, you take carbohydrate reaction here too. Then in your biomass reaction, you won't have macromolecules, but you will have all the building blocks, amino acids, fatty acids, nucleotides, etc. together. So this is the second alternative I was talking about. So you will have a biomass reaction from not macromolecules, but from building blocks within a single reaction. That's the same. I mean, you can prefer this one or you can prefer this one. They will give you the same results when you do simulations. Third alternative. This third alternative is not used nowadays. It was used in 90s when these metabolic network modeling papers first appeared in literature. So here, the models were much more simple. The metabolic network reaction list was more simple in the models in 90s. They did not include biosynthetic reactions most of the time. So this can be a little bit tricky. Just try to follow, and I will go slowly. So for each building block, building blocks are made up of metabolic precursors and ATP, NADH, and NADPH, right? So we say that metabolic precursors, ATP, NADH, and NADPH, produced in the central carbon metabolism, are used to make building blocks in the anabolic part of metabolism. So we can, for each building block, we can make a list of required precursor and required cofactors and ATP. For example, for alanine, you need 1 pyruvate and you need 1 NADPH and 1 NH3. Or for leucine, here, you need 2 pyruvate, 1 acetyl coenzyme A, and you need 2 NADPH, and you need 1 NH3. Or for valine amino acid, you need 2 pyruvate, you need 2 NADPH and 1 NH3. Again, a table from Metabolic Engineering Principles book. So these early models, they did not include the biosynthetic reaction in the model, and they rather considered this table, how each building block is formed from the metabolic precursors and ATP, NADH, and NADPH to make a smaller size model. And this was the trend in 90s, as I said. Nowadays, almost nobody uses this approach. Again, for other building blocks, such as nucleotides, you can write them in terms of required precursor metabolite and required ATP, NADH, NADPH, etc. And also for lipids, also you can write them in terms of precursor metabolites and required ATP, NADH, and NADPH. So just to show you quickly what this means, I will go back a little bit. Yeah. For example, leucine is a building block, right? And this is how leucine is formed. It requires 2 pyruvate It requires one acetyl coenzyme A. It requires one NADPH, and it requires one glutamate. But glutamate is made up of one alpha ketoglutarate and one NADPH. So one alpha ketoglutarate and one NADPH. So in those early models, rather than writing those reactions for the formation of leucine, they considered the requirements in terms of ATP, NADH, NADPH, and precursor metabolites. So in this case, for leucine, we need 2-pyruvate, we need one acetyl coenzyme A, we need one alpha ketoglutarate, we need 2 NADPHs. So remember this biomass reaction from building blocks, this many leucine plus this many alanine plus this many phospholipids, etc. So rather than writing biomass reaction in terms of building blocks, this approach writes biomass reaction in terms of precursor metabolites. So let's go back there quickly. So rather than writing, for example, where is our leucine here? Leucine here. Leucine requires one alpha ketoglutarate, 2-pyruvate, 2 NADPH, etc. So all those requirements are multiplied by this coefficient, and rather than leucine here, they write alpha ketoglutarate, pyruvate, NADH, NADPH, etc. So each building block is written in terms of its required precursor metabolites. And at the end, we will have biomass formation reactions in terms of precursor metabolites and required NADH, NADPH, and ATP, etc. So this is the third approach, as I said, used in 90s. And by using this approach, by compiling catabolic and anabolic reactions and by adding the related biomass reaction, and also by noting the genes that control the enzymes of those reactions, you make a list of reactions which are referred as genome-scale metabolic networks. We will talk about this in a very detailed way in the coming weeks, but some example from literature. For yeast, for example, the first genome-scale metabolic network was published in 2003. We already saw some tables from this paper. So the initially published genome-scale metabolic network had about 900 reactions between 750 metabolites controlled by about 700 genes. And currently we have a much larger genome-scale metabolic network. With the improvement in our scientific knowledge, those genome-scale organism-specific metabolic networks are updated in each couple of years. For human, for example, they have published the first human genome-scale metabolic network in 2007. It included about 4,000 reactions between 1,500 metabolites, and those reactions were controlled by about 1,500 genes. And the most recent human genome-scale metabolic network was published this year. This is 2020. There's a mistake there. And this recent model includes 13,000 reactions between 8,000 metabolites, and the reactions are controlled by about 4,000 genes. So you can use these huge amount of reactions to make simulations about the phenotypic behavior of your organism of interest. And there are databases to get those reactions. You can use Cake, Biopsych, and other pathways to see organism-specific reactions and to combine them together for your modeling goal.